everyone. In this lecture, we will talk about the pathogenesis of viral infection. Viral pathogenicity is the result of the interaction between virus and host. The adsorption of viral attachment protein to host cell receptor determines the tissue tropism of virus. The factors that determine the viral transmission, multiplication, dissemination, and development of disease in the host involve complex and dynamic interactions between the virus and the susceptible host. Viruses cause disease when they breach the host's primary physical and natural protective barriers, evade local tissue and immune defenses, spread in the body, and destroy cells either directly or via immune and inflammatory responses. The ability of viruses to cause disease can be viewed on two distinct levels: the changes that occur within individual cells and the process that takes place in the infected patient. Let's look at the direct interaction between viruses and the cells first. To produce disease, viruses must enter a host, come in contact with susceptible cells, replicate, and produce cellular injury. Interaction between virus and host cell results in the following outcomes. Cytocidal infection occurs when the target cell is lysed due to the replication of virus. In this case, viruses replicate and release from host cell in a short period, resulting in the death of the host cell. The main mechanisms of cytocidal infection include several aspects. Viral infection inhibits the biosynthesis of host cells, or influence of virus on cell lysosome increases its permeability and eventually leads to cell autolysis. Viruses damage the organelles of the host cell because of toxic effect of viral proteins. Some virus infection can cause nonspecific injury of cell nucleus, endoplasmic reticulum, mitochondria, and so on. The morphologic changes in host cell caused by viral infection were known as CPE, cytopathic effect, including cell swelling and rounding, loss of adherence, cell fusion, and cell lysis. As the picture shows, the left is normal cells and the right is the virus-infected cells. We can see the significant changes in the morphology of the cells. This kind of infection occurs mainly in naked viruses. This picture shows the cytopathic effect of enterovirus 71 and HSV in cell culture. Note the ballooning of the cells. This picture shows the syncytium formation in cell culture caused by RSV and measles virus. Viral infection can also result in steady-state infection. Steady-state infection is usually caused by enveloped viruses, whose progeny viruses release from host cell by budding. This process is very slow, so host cells are not lysed or killed in a short time. Steady-state infection can cause cell fusion and produce multinucleated giant cell. Cell fusion is one of the ways in which enveloped viruses spread. Steady-state infection may also lead to the appearance of virus-specific antigen on the host cell membrane, so that the immune system may recognize it and attack the infected cell. Therefore, the infected cells eventually die because of the continuous viral replication and the attack of immune cells. Infected cells frequently contain inclusion bodies, which are discrete areas containing viral proteins or viral particles. They have a characteristic intranuclear or intracytoplasmic location and appearance, depending on the virus. The inclusion body can be seen under the light microscope. One of the best examples of inclusion bodies that can assist in clinical diagnosis is that of Negri bodies, which are eosinophilic cytoplasmic inclusions found in rabies virus-infected brain neurons. Another important example is the oil's eye inclusion seen in the nucleus of cytomegalovirus-infected cells. Electron micrographs of inclusion bodies can also aid in the diagnosis when virus particles of typical morphology are visualized. Some virus infections delay early apoptosis, which allow time for the production of high yields of progeny virus. Additionally, some viruses actively induce apoptosis at late stages, which facilitates spread of progeny virus to new cells. For example, HIV induces the apoptosis of CD4 positive T cells, leading to cellular immune deficiency. Some DNA viruses or retroviruses can integrate the viral genome into cellular DNA, which is called gene integration. This gene integration can cause malignant transformation of host cells, which is characterized by unrestrained growth, prolonged survival, and morphological changes, such as focal areas of rounded, piled-up cells. This process is called viral transformation, and these viruses are oncogenic viruses. The most common oncogenic viruses and corresponding tumors are as follows: HBV and hepatocellular carcinoma, HCV and hepatocellular carcinoma, HPV and cervical carcinoma, EBV and Burkitt lymphoma and nasopharyngeal carcinoma, HHV8 and Kaposi's sarcoma, HTLV and human T-cell leukemia. The signs and symptoms of most viral diseases undoubtedly are the result of cell killing by virus-induced inhibition of micromolecular synthesis. Death of virus-infected cells results in a loss of function and in the symptoms of disease. However, there are some diseases that are not caused by the virus damaging or killing the infected cell. In these diseases, destruction of the infected cells by immune response may have severe or fatal pathological outcomes. Pro-inflammatory cytokines, antigen-antibody complexes, complement activation pathways, CD4 positive T-cell induced delayed hypersensitivity, and CTL-mediated cell killing contribute to virus-induced immunopathological injury. Viral infection may cause immunopathological reactions, such as hypersensitivity. After viral infection and replication, some new viral antigens appear on the surface of the host cell membrane. The specific viral antibody can recognize and react with these antigens, and the host cell may be damaged in the presence of complement. In some viral infection, immune complexes are formed by interaction between viral antigen and specific antibody. The degradation of the immune complex may activate complement, contributing to the immunopathological injury. Virus-infected cells may also be lysed by cytotoxic T lymphocytes. These pictures show different types of hypersensitivity. Viral infections in several instances can suppress the immune response. Immunosuppression can be achieved either by direct viral replication or by viral antigens. Some viruses specifically infect and kill immune cells, such as HIV. Viruses infecting either CD4 positive helper T cells or antigen-presenting cells cause immunosuppression. Viral gene products can cause immunosuppression by stimulating pro-inflammatory cytokines. Viral infection may include an autoimmune response because the viral protein resembles a host cell protein, induces a phenomenon called molecular mimicry. Both viral epitope-specific antibody and T lymphocytes may react with cognate epitopes on the host proteins, which may elicit an autoimmune response. Okay, that's all for the pathogenesis of viral infection. Thank you for watching and see you next time.